Good morning, doctor. We are from Group 6. Today, we would like to discuss about our assignment 3, and the topic that we choose is about genetic.
So for the first part is about the introduction. Genetics, also known as science of heredity, is the study of biological information and how this information is stored, replicated, and transmitted and used by subsequent generations. The study of genetics can be subdivided into three main areas, which are transmission genetics, molecular genetics, and population genetics. Genetics is the branch of biology that deals with the study of genes, heredity, and the variation of traits in living organisms. It is a fascinating field that allows us to understand how the characteristics such as eye color, height, and even certain diseases are passed down from one generation to the next. As the time goes by, people always had an interest in genetics and its effect on their physical traits and inheritable characteristics. As organisms reproduce, new organisms may inherit some of their dominant traits and lose their recessive traits.
So, I believe that now we have a brief understanding in the study of genetic after the explanation from Nurul Amira. So, now I would like to continue by talking about one area of mathematics called linear algebra is very useful and helpful in solving a number of real-world issues, such as those issues in the study of genetics. So in genetic, linear algebra is an essential thing for the tasks like understanding the inheritance pattern, modeling the biological system, and also interpreting the genetic data. So hence, the linear algebra concept can provide a tool for us to manage and interpret the complex data structure inherent in the genetic research also.
In this group assignment, we will explore how the concepts in linear algebra can be applied to genetics. We will go through the fundamental applications of linear algebra in genetics, such as how matrices, vectors, and linear equations play a crucial role in modeling, calculating, as well as interpreting genetic phenomena. We will also go through three main real-life examples of the topic in use, which are effect of reproduction on host color, genotype distribution of an organism, and genome sequencing.
Okay, now so we will go to the second part, which is the linear algebra prerequisite.
So for the first linear algebra prerequisites of the color of genetic traits of an organism, there are three. For the first one is vectors. The genetic distribution of horses is represented as a column vector. For example, the initial state vector shows the proportion of genotype, which are double capital capital E and small e, and double capital E. At the start, at 0 is equal to proportion of double capital E, proportion of capital E and small e, and proportion of double small e. And second is matrices, where the transition matrix summarize how genotype change due to reproduction. For example, the rows represent the probability of genetic outcome for specific parent pairings. In this case, it's a matrix where each column correspond to one of genotype. So E equals to double capital E probabilities, capital E and small e probabilities, and double small probabilities. And the last one is metric multiplication to find the genetic distribution for the next generation. So we use current distribution vector by matrix x n plus 1 is equal to e times x n.
So now we need to see the prerequisite for the second one, which is the genotype distribution of an organism. So the concept we use is the matrix diagonalization. So it is the process of converting a square matrix into the special matrix, which is the diagonal matrix. So a square matrix is said to be diagonalizable if it exists in this the form where D equal to the inverse of P multiplied by A and matrix P. So the common form of the diagonal matrix is as follow, where the lambda, all the lambda is the eigenvalue of A, and we need to use the concept of this diagonal matrix for the next part. So the previous slide we talked about the concept of diagonalization of a matrix. So now we need to use the concept to compute the power of a matrix, which is simplified in this slide. So we need to transform the d to the power of k equations into the a to the power of k by making it is the subject of the equation, because the a to the power of k is our interest that we need to find. So here we also need the method to find the inverse matrix of the P. To find the inverse of an invertible matrix, we need to find a sequence of elementary row operations, which is simplified in the notation below there. we reduce the P by using the elementary row operation into the identity matrix. So at the same time, we also reduce the identity matrix to the inverse of P. So by calculating the power of the matrix, we can investigate the probability or the possibility that an inherited trait is passed down across the generations.
In order to understand what is genome sequencing, we need to understand the topic matrices, such as understanding of matrices as representations of data and representing genome sequences or alignments as matrices by converting the fragments of DNA into a matrix. Besides, we also need to know about matrix operations and properties, such as finding the transpose of a matrix, as well as matrix multiplication of matrix and its transpose.
Okay, so now we will continue to the third part, which is about the real life or practical example.
So for the first real life example is about color of genetic traits of an organism. Concept linear algebra is applied to predict different outcome of color of genetic traits among host hair color. Is demonstrated by Armin Moshrafi, Sarah Rogers, Megan Wolf and Jason Zambrano in 2012. So genes inherited from biological parents are expressed in specific patterns, influencing various traits such as color of organism. Most genes occur in pair, one inherited from father and another one from mother. So genotype refers to genetic blueprint of organism encompassing all gene passed from one generation to the next. The distribution genotype within a population represent the proportion of or frequency of different genetic variation. So for the methodology, let capital E as the dominant allele and small e as the recessive allele. So when crossing animal, offspring get one gene from each parent. So double capital genotype horses are dominant color, so bay, and capital and small e genotype horse are hybrid horse with a dominant color, bay, and double small e genotype horse are recessive black. So for the first case one, so we consider crossing the double capital E with another double capital E. So probability of getting double capital E, capital E and small e and double small e will result in 100 vector. So for the case 2, double capital E with capital E and small e bay house, so probability getting a 1/2 vector. And case 3 consider crossing capital double capital E with double small e, so probability getting 010 vector. So here are the problem. Horse start with two color, black and bay. Black horse has recessive gene, while bay horse has either dominant or hybrid gene. So within the first round of combination, black color disappear and all horses will have bay color, where is a horse with a black mane, tail and a brown coat. So we want to identify the next generation of color of the horse from the methodology before. So all the cases we will times with the X naught. X naught is the three possibility of the allele which likely to occur, hence the initial matrix at 0, where the entries represent double capital E and small e, and double small e genotype, and Xn, where n is the number of the year, by using the formula of Xn equals to E Xn minus 1. So we use this formula to calculate the next generation.
So now we will go through the second real life example, which is about the genotype distribution of an organism. So the concept of linear algebra is applied to find the genotype distribution of an organism. For example, the Jared Gilcam in 2001 used the concept of linear algebra to study the genotype distribution of the flower population in the N generation. So the method we use is by assuming the starting genotype distribution is the X naught and let the matrix A to represent the genotype transition matrix for the specific population. So after the year, the genotype distribution is denoted by x1 is equal to the A times with the x naught. So the second year, the genotype distribution also can be defined by x2 equal to A multiplied by the x1 and also equal to A multiplied by A x naught. Then this is equal to A squared x naught. And we also can compute by the same method to find the x3, x4 until xn equal to A power of n x naught. So now we will go through a specific example of problem which that a farmer has a large population of plant with three possible genotype and he want to know the probability of the possible genotype of the offspring produced after a certain number of generations. So the first step we need to do is the crossbreeding. After the crossbreeding, we can get the probability, and then the probability we can write it into a matrix form, which is the matrix A, which represent the genotype transition matrix, and we can compute the x naught by assuming the genotype was present in equal proportion. And the X1, X2, X3, X4 are also can be computed by using the formula from the previous slide. And now we will introduce the diagonalization method. And as we know, the xn equal to a to the power of n and x naught. And the a to the power of n can be written as p multiplied by the d to the power of n and the inverse matrix of p, where the n equal to 1, 2, 3. And now we can obtain the eigenvalue and the eigenvector, which are denoted by the lambda 1, lambda 2, lambda 3, and v1, v2, v3. Therefore, we can obtain the diagonal matrix D is this one, and the P, the matrix P is this one. And then we need to compute the inverse matrix of P also by using the elementary row operations method. And now we can combine the two equations which are highlighted in the blue color to obtain the new equation, which is the x n equal to P multiplied by the D to the power of n and P inverse and x naught. And then we compute it, we can get the x n, which can be used to find the proportion of the population of the plant after the n generations.
Before we move on to implementing genome sequencing using linear algebra, let me explain some key terms that will be used later on. First of all, nucleotides are the basic building blocks for DNA. There are four of them, namely A, G, T, and C. A stands for adenine, G stands for guanine, T stands for thymine, and C stands for cytosine. Fragment is a piece of DNA that can be created in a lab or occur naturally. DNA, or deoxyribonucleic acid, is a molecule that contains the genetic information for an organism to develop and function. Genome is the complete set of genes or genetic material present in a cell or organism, while genome sequencing determines the complete DNA sequence of an organism's genome. It identifies the order of nucleotides. Next, I'll be explaining the method on how to implement genome sequencing using linear algebra. First, we convert fragments into row vectors and arrange them into a matrix. Then, we find the transpose of the matrix and perform matrix multiplication F multiplied by the transpose of F. Then, we find the overlap scores of off-diagonal entries and draw a graph to represent the fragments and their overlap scores. We will also find the fragment with the highest total overlap score to start the sequence. We will also use the graph to trace out the sequence of fragments and finally merging the overlap regions of fragments to form the complete genome. Now we are given a problem with four fragments and are asked to determine the optimal fragment order for genome assembly. So the solution is, first of all, sequences are converted into row vectors and are arranged as rows in a matrix for comparison, where rows represent fragments and columns represent nucleotide positions, such as the diagram shown below. Then we will use the matrix multiplication F multiplies by the transpose of F to calculate pairwise overlap scores. This measures the similarity between each pair of fragments. Then from the matrix multiplication, diagonal entries show the self-similarity of each fragment and these values are ignored, while off-diagonal entries represent overlap scores between fragments, as shown at the diagram below. After that, we will draw a graph. Each fragment is a node, and overlap scores form the weights of the edges between nodes. And after proceeding to the node, it cannot return back to the previous node. To reconstruct the genome, we need to start with the fragment having the highest total overlap score. Fragment 2 is chosen because it has the highest overlap score of 86. Based on the graph, we now move from fragment 2 to fragment 4 as it has the highest overlap score. Then we move from fragment 4 to fragment 1 as it has the second highest overlap score. And finally, fragment 1 to fragment 3 as it has the third highest overlap and is the remaining overlap score. Therefore, by using the order fragment 2, fragment 4, fragment 1, fragment 3, overlap regions are merged to form the complete genome, and the resulting genome is shown below. Before we end our presentation, these are the references that we use during our presentation. Last but not least, thank you, Doctor, and we hope to see you again in the future.